Okay.
You can actually park your, you can go into your brow touches.
Yeah.
Yeah, just don't touch the Norbit.
Yeah, that's what I was thinking. I can hover here. That's fine. We don't need to. We don't need to break the Norbit.
Right.
You can hover. That's fine. It's just a quick.
Can we get
snap.
a half zoom?
You bet.
I like this brittle star on display.
Yeah, wow.
There you go.
Not
Great
afraid
The
of
way
it.
he
capture.
twirls his arms around the branches of the coral.
Yeah, they are definitely holding on.
Kind of reminds me of a Christmas ornament on a Christmas tree.
A little bit, right?
This is interesting. It's probably so we have a colony, probably a Hemicorallium right here that has been completely taken over by a white Zoanthid. The polyps you see are not the polyps that created the skeleton. And I don't see any live Hemicorallium polyps, so this one's been completely taken over.
Wow.
And it looks kind of palish pink, which is probably why it's starting to lose some of its, maybe some of its pigments out of its tissue, so it's not really as clear. But there's a really interesting observation. Usually you see them in the process of being taken over by their associate. Thanks, we're all set.
Okay.
Front row.
Come wide.
But this one's completely taken over the individual.
Little dust storm there.
Little blizzard.
And you illustrated another fantastic feature about the coralliids is that they're inflexible,
Yeah.
which helps us confirm their ID.
So Steve, when they're taken over like that, does the branch structure stay healthy and alive?
Come up. I've got some clear water.
Okay.
For some time,
Come up and ahead. There's
but not forever.
Roger.
Okay.
Bring it
The
up at the top
zoanthids
to
don't
look
have
at.
the ability to calcify, so pretty much what's there is what's there.
Gotcha.
And to make matters worse, there's a type of polychaete worm that lives inside the skeleton of precious corals that kind of undermines it. It burrows within the skeleton and weakens it in some way. And sometimes that can cause failures of the colony.
Tilt your camera down a little, Chris.
Camera down.
What's the time span on the takeover?
You're looking up into your light.
A long time.
I always look at that and we like describe it in these this is the parasitism. You're like,
going on here.
this is a total horror movie if you were a coral. Like, this is like a zombie animal living on the skeleton of another animal.
Just below you.
Corals have it rough, yeah. Everything wants to eat them.
Yeah.
They're almost like, the plants or the trees of the deep sea.
Yeah.
Sponges as well, but everyone wants to eat them.
Well, they're just hanging out, like little snacks.
And kind of the top of the food web here are like sea stars
Yeah.
most of the time.
No.
Not many things will eat the sea stars.
Anytime we have kind of this like tilting angle like this, it really throws my brain off. I'm like, how did the ROV like get tipped over? When you get these diagonal, doesn't that look so?
Yeah, it does look a little
yeah.
I'm just going to turn auto head off and let you take the leash.
Right on. I'll come up too.
This is my favorite kind of terrain to explore when you're at a shallower site, like a shallow atoll or island, like the part where it just goes vertical, because you get all sorts of really interesting flow conditions, and corals love the vertical faces because they're relatively stable compared to kind of the top or rubbly layer underneath.
Why don't you come back to your left a little bit, get back closer to Argus.
Okay.
So when we say carbonate cap, what does that mean?
So there's pulling on your tail.
So I guess we can start with the first word, carbonate.
Stay on the cliff top
I guess
there. But
more accurate
I see what
would
you're
be
saying.
calcium
Yeah.
carbonate, but
Oh,
Shrimp? Hello,
oh.
shrimp.
wow.
Shrimp
shrimp.
attack.
Run away!
Cool.
Carbonate is a compound, CO3, and calcium carbonate adds calcium in there. That's calcite. Coral are actually a polymorph of calcite. called aragonite, and they become calcite when they die and they lithify into a sedimentary rock, a carbonate or limestone you could call it. We call it a cap because it is a cap on top of the volcanics below it. And it is a cap because
Bring your head to the right a little more, Gibson.
when
and
the
look down.
volcanic island was near the surface, the
Trying, Dennis. Just
coral
keeps
were
twisting
there
back.
for a long time, and
What's that?
Keeps twisting
tons
me
and
to
tons
port
of them were
side
there,
here.
and
Yeah, come down.
they
come
eventually
down 5
lithified
meters.
into this giant
Coming down
layer we're
5.
seeing right now.
Yeah, we might want to move the ship a little more south too,
There we
because
go.
they were getting pulled up the cliff.
Yeah, we can do.
Yeah.
And what are some of these other corals that we're seeing along the ridge of this platform?
Yeah, like that, I guess.
So there's a
20
mixture of
meters,
crinoids
135,
here.
I would say.
Got a couple different species
that'd
of
be all right.
Anthomassus in the area, Bathypathes, black corals,
Yep.
umbellapathyes. Got at least one species of Narella, Candidilla giganteaus present here.
No, 135.
We've also got this yellow plexaurid. What else we got? Some Hemicorallium, paragorgia, Chrysogorgia. We saw there was a
You're
bottle
basically
brush
moving,
back there a bit.
Atalanta right towards her.
So what it is that, 10 species.
Amazing.
Yeah.
Nice.
So that's just in this one small area.
Beautiful.
Fine, I'll jump in, Chris.
Yep.
Ready?
Well done, Chris. Yay, guest pilot, Chris.
So Brandon, do all gyotes have this calcium carbonate cap on it?
Do all islands?
Gyotes.
Yes.
Oh, geos.
Mhm.
Yeah, that's part of the definition of a geo, or at least a geologic definition, is they have to have a carbonate cap on top.
Beautiful.
Thanks, Chris.
Nice work. Yeah, so just looking at more history of this feature, right, after the eruptive stage, or
Right, island stage,
yeah.
you could call it.
yeah, in the island stage, you have corals attempting to grow and keep up and stay up in the photic zone, in the sunlight zone, and growing basically straight up, right
Yep.
on top of each other. Hard corals, snorkel corals, the corals we,
Right.
most
Shallow
people may be
corals.
more familiar with. And
Forgot
growing and growing and
Metallgorgia
growing
too. Saw that.
until
We're going tail
it
tail then.
subsides.
Subsides, yes. The volcanics will erode because of wave action. But corals can beat that erosion a good amount of the time, but eventually they
Doesn't
fail,
look like she's moving 135.
and they become a cap.
As
And
they sink into the sponge cake jello of the coast.
Yes, precisely.
Move her south 20 meters. Due south 20 meters, yeah. You can come down 5 meters there. Copy that, Gibson?
Come again, Dan.
down
Science
5
Steve
meters.
fans in the house.
Occasionally they let me go home. And then guess what? I'm back on the computer, joining from my
Surprise!
home lab.
It never ends, Steve.
We like you. We'll keep you.
But as soon as the ice cream's gone, I'm off the boat.
It is almost Sunday.
Every day is Sunday for me.
There's some pronounced layering right here too. Pretty nice.
That is nice.
Yeah.
So why doesn't this look like a shallow coral reef if it is a
Can't get
carbonate
around
cap?
That's all right. You can stay tail to tail.
Yep. Roger that.
Millions of years of lithifying
Okay.
makes stuff look different. Would be my estimation of it.
Does manganese settle at the same rate onto basalt and onto carbonate rock? Like, is it an equal opportunity blanket, or?
It will accrete onto both of them. I'm not sure if it changes because the limestone's biotic.
Mm-hmm.
Look straight down.
Looking straight down.
Probably not. I would say it's probably the same, but if somebody else knows better, please.
See, that's the one we saw
So
before, is
can
it?
You're a
this-
resident geologist.
I'm also
What you say
still
goes.
quite a novice.
There we
Fair.
are.
So can this manganese crust, can it form on coral and sponges over time? Or does it have to wait, do coral and sponges have like a defense mechanism or a way to get rid of it?
So we've seen it formed on there, on this dive, or on the last dive. And Steve is talking about how it's unclear how quickly it is. It must be quicker than how fast it grows on the rock. But yeah, it will grow on the coral, at least at the base.
Yeah, we picked up that skeleton. The
Is it a
I didn't
sponge
see it when it
skeleton?
came back up. I think sponge skeleton that was coated in ferromanganese crust last year on, I believe it was on Expedition Lu'ua Ea Hike Kapapaku in Papahanaumokuakea. We found a beaked whale skull that
What's
had
that,
been
Chili?
totally coated in ferromanganese
Floating
crust,
right
which
at the
was
bottom of your
really cool.
screen.
What's that, Steve?
There's a red jelly down
So
there.
you knew
Yeah.
better than me.
Right there.
Hello.
Alright, zoom in a bit on that guy.
Yeah.
See if we can catch him on the fly.
Oh, look at that.
Oh, it's a
Thanks.
Siphonophore.
Sea dandelion.
Mm-hmm.
Yay.
Oh, they're beautiful.
Should be good to come down a couple more meters now.
So
Me?
are these fairly common at this
Altimeter's
sort of
reading
depth?
7.
Because I haven't really seen one ever.
They can be found through
Wide.
a very wide depth range. They're more common, shallower, like most things. But yeah, we can find them. Sometimes they're attached to the substrate with anchors. Sometimes they're free swimming like this. They have ways of moving about, those kind of pumping zooids create a small current that they can use to propel themselves.
This is like the ultimate teamwork animal. It's genetically identical zooids that are differentiated into different body parts. So some will be locomotion cells, some are digestion cells, some are anchors, but they're all genetically identical before they get jobs.
So
Get a job.
are these sort of jellies? Are they filter feeders? Do they hunt? How do they eat?
So they're primarily suspension feeders. They have special zooids that are used in
Dude,
particle capture or
2135.
defense. They sting their prey and then digest them.
How about more southish, maybe 150, something like that. 150.
And so what do those
Yeah.
jellies primarily feed on?
Popping it up.
Anything that's in the water column, usually. So planktonic organisms.
Yeah.
Does that include like coral and sponge, or just?
No,
Sort of.
yeah, they will often get caught on corals and sponges if they flow into them, and they'll probably be consumed by the coral. I don't know if they'll be able to fend off a whole colony. Sometimes you find gelatinous plankton just smushed into a colony, and they'll probably get eaten
Could eat them.
over time.
So cool. Yeah, Siphonophores relatives to corals, anemones,
Yep.
jellyfish. In the group Cnidarians.
All right. Not to be a total downer.
Uh-oh.
This is cool, but can we might have to break off here and kind of head upslope towards 11, just to get kind of up on top of the
Brittle
summit
star
here.
coming down the left side.
Oh yeah. jumped.
Oh my
it might be
gosh.
a that might be a squat lobster with
Okay.
its pinchers
Is
out.
that how they go down?
Oh,
They're just free fall.
Stauropathes here. So yeah, there's
Creating
15 to 20 species here in this small area. You think we could do a Niskin bottle here?
Sure.
Kind of, no more than a couple meters off the wall if you can.
We are currently about 2 meters off the wall.
Yeah, 15 to 20 species in this area of different kinds of deep water corals. Not as many sponges, so sponges have gone away. Corals have emerged in this area. The mustard color coral in the center is Stauropathes, S-T-A-U-R-O, pathes.
So Steve, we're going to do an environmental DNA sample here, a water sample. What DNA are we capturing? Like, what is coming off of all of these animals for us to be able to detect them from a water sample?
So when organisms release mucus, fecal material, they will often have DNA that is associated with that. Usually, cells that might have sloughed off in that process, and that's something that is free floating in the environment in the water column, and it oftentimes, until it gets degraded, it's a fairly stable molecule, so it can free float around for quite a bit. It turns out we can detect that DNA if we capture it, filter it, and sequence it, a fairly short distance away from the colonies. We're looking specifically for corals in this case, or soft octocorals for the most part, but other things as well. We're specifically looking at mitochondrial DNA for corals, since it seems to be the most informative at delineating species. So most people might not know you have a couple different types of DNA in your body. You have nuclear DNA and mitochondrial DNA. Mitochondrial DNA is usually passed down the maternal lineage. So for corals, it turns out that mitochondrial DNA is very useful for separating one species and another.
Let me chase the wall up for, it's going to end here in 10 meters.
So that was Niskin 5, right?
Oh, I love these bright red crinoids.
Such a nice flash of color in all the ferromanganese crust.
Yeah, I think they look like Gerber daisies. So you'd like get in a bouquet.
Flowers.
Mm-hmm.
Make a coral sponge bouquet.
Mm-hmm.
Okay, you can head up.
The bathymetry on this area would be quite pretty at a higher resolution.
Definitely.
Big wall face.
It's features like this that always make me want to like 3D print the bathymetry. Like I want to have it,
Oh,
you know,
that
as a
would be so cool.
manipulable, right? To be able to see these ridges. Like often they incline like the low slow slopes. You're like, okay, you know, I kind of have an idea of that, but particularly geos and then these like, I think of them kind of having like buzz cuts, you know, when they go like straight up right at the top and with this super sharp edge.
Gives me a great business idea if this science thing doesn't work out.
Hey,
Climbing gym based on 3D bathymetry of geo
yeah.
X in the Pacific.
What would you
Sounds
call
actually
it?
kinda cool.
Oh, that's a good question.
It would probably be some sort of epic pun.
Gotta think of it by the end of the watch.
For the Venn diagram of ocean explorers and rock climbers, which I actually think has a pretty big overlap,
Yes.
Yeah.
maybe folks can contribute their ideas in the chat and
Start engage checks.
Roger. You can come down,
give folks a few
Okay.
hours and we'll
down 10.
see what
Coming
they
down
can.
10.
Give you a heads up whenever you can get there. We'll do a quick zoom on that
Yeah,
thing coming into
headed there.
view.
Want to go tail here?
Actually, that's good on the delta. You can hold what you got. Let's just go tail to tail. Yeah,
Okay. Auto heads off.
roger. And then it'll say, I told you so, but I waited too long to move the boat. Nothing.
Something free floating to the right of it.
Okay, you can
It's
zoom
just such
in
a
on that, right?
terrifying view.
Yeah, I actually clinched a little.
Yeah. Let me bring a little more light back into that. Pan down.
Yeah.
Tilt down.
Are these snake stars or is that another one of those common names that is too broadly applied?
Too broadly applied. These are, yeah, a different family of
Okay.
brittle stars. I think these are ophiocanthids with the spines.
All right. Do snake stars have no spines?
I think they have more smooth arms.
Okay.
Yeah.
And are we seeing two different types of brittle stars here, or because we see like the red ones and then the little white one down below? Is that a brittle star, or is that just a younger,
have any more
not
zoom?
mature?
Yeah, there it is. Oh, this was coming in. Yeah, I think that's a different kind.
A little one there.
Oh, there was also one on the rock below.
Do we know what kind that is?
Processing.
That's max. So I believe this is in the genus Narella. Although I can't put it to a species now. Do you have time to take a snip of this colony?
We do.
And we'll have to go in one of the bio boxes because I don't know if it'll slurp.
Okay.
Anything can slurp if you try hard enough. The question is if you want it intact.
We have jars. Oh, you don't think it'll go through the tube?
Yeah, it depends on how much of a branch we get.
We could put it in with the cucumber in the front.
Yeah, we have 2 cucumbers
yeah.
in lambda, and the rest of the bio boxes have rocks and a fossilized coral in it.
They are very floaty, those cucumbers.
Yeah.
So if you can do it without being able to open the box enough
I think so, yeah.
that the sea cucumbers can't get out, then we can try. But
Roger that.
it's a delicate thing.
The sea cucumbers in the close or further back into the vehicle?
They're in the 4 lambda bio box.
What's on this side?
Rock. 10 centimeter rock.
Yeah. If you can put it in the forward bio box,
Yeah, we can.
the right starboard partition is preferred, just so
Okay.
it doesn't get all mucked up by the cucumber goo.
Roger.
Oh,
Yep.
see you later.
That's what they do.
Freedom!
Bye-bye.
Freedom!
So this species of Narella, to the best of my knowledge, has not been sampled in this area.
We hit 5 on the craft camera.
There are some guides place it as Narella question mark, which is another point for supporting its collection in this case.
Who is the Narella specialist? Like, who might the sample go to?
Well, I'm gonna look at it first.
Okay.
But
Like me.
yeah.
Awesome.
So there's not many. It's like a handful, maybe 2 or 3 people that really specialize in primnoids. 2 folks that I've worked with in the past are Dr. Steve Cairns at the Smithsonian Institution, National Museum of Natural History. He works on primnoids widely, and then as well, there's a scholar in the UK, Michelle Taylor, who works on the genus Narella as well as Thuarella, among others.
Awesome.
But both of their publications have produced some pretty awesome keys that allow us to more easily identify primnoids. If you can get a branch that's undamaged, that would be ideal.
That top left one there that I'm after. Any circle one you want.
Yeah, maybe this one, but in like the back. This one right here looks really might be damaged.
Zoom in there
Yeah.
for us.
So either
And get the bottom
here,
one.
yeah, the bottom one, you
Yeah.
got a couple of branches, or even like this one would be okay too. We don't need a huge piece.
Don't want that one. This guy here.
Yeah, that would be better.
There's a good angle here.
Yep.
The brittle star landed
Yeah,
on our front
the
porch.
DSC cam has another good view.
Oh yeah. Too bad it's not on a tilt. Should be a requirement for ROV cameras. Okay, I'm gonna go right in there, Steve, and snip that. You all right with that?
So worth pointing out that this colony overall doesn't look super healthy. It's missing a bunch of branches. Unclear what could have caused the damage, but nothing actively preying on this now. Even the ophiacanthids, while they may kind of frustrate the growth of the colony, they don't actively predate upon this coral. That looks like a great piece.
Got a little brittle star friend on there too. Bonus
Yeah.
points.
Yep.
Associates
Associates are also helpful because sometimes some associate species have very high fidelity, which means that they help us identify certain species because they
Zoom in
only
there
occur
for a
in
sec if
species.
you want.
Sure. Yeah. All right.
Okay, if you go wide now.
I still think
Where do you want it? Any?
if you go forward, you can't open the drawer all that much.
Yeah, no.
But the starboard side and the forward box, if you want to go forward.
Roger.
Starboard box would just be a safe option too.
You want to go in the tool tray here, or you want the bio box?
We're gonna go on a tool tray, but don't open it yet.
Okay.
Think he'll float or you think he'll sink, Steve?
It will sink.
Sound pretty confident.
100%.
100%. Okay, let's give it a bump. Open. Yep. You see something start to float out, just go ahead and close it.
One more. Oh.
Yeah. Close it.
Oh.
Close it. There.
Was that just a piece of it, or was that?
I think that was a piece of it, but
You want me
yeah,
to close it all the way?
that wasn't the gut piece, so that's not the important piece.
No, bump it open a little more. See what
All right.
happens.
Sorry, Liz.
Uh-oh.
Oh.
Yeah, close it, close
Let's
it,
close
close it. Close it, close it.
I'm closing as fast as I can.
Wait, no.
All right, we're gonna need a rescue.
All right, we can go to the starboard box
Yeah, hold on.
if we can coax that back in there.
Yeah. Okay, I'm gonna float up and then, hold on. Okay. Close it.
Nice.
I guess I won't open that box.
That's amazingly floaty.
Wow.
It
Didn't
really.
Did
No,
not
we're gonna go to the
expect that.
starboard box.
I thought like neutral
one of
was
these
kind of what a
compartments.
sea cucumber would be.
Yeah, well, one of them. They're all rocks in here,
To the
so
left
there's
a little
nothing
bit
that would be.
forward.
You can always double up,
Very
unlike.
I think it's a
Specimens.
conscious,
And it
a
won't break
conscious
the
movement.
coral.
No.
Mm.
Like they're purposely trying to escape.
I'm gonna go ahead and look
I
to the
don't
left
think-
and
I
get
don't
her
know.
can
I
be
thought
in.
she said that.
We're gonna float here
Was
while we're
the-
doing this.
Oh, I'm not sure. I
it's
missed
like-
that one.
Yeah, that they're swimming away.
It's a really, we see it with all kinds of samples, kind of like opening the box creates kind of like a whirl of water going into it. You know, it's a closed container otherwise, so
That's a
very
good shot
possible
right
it
there.
just kinda-
You'll see us open the box.
pushed it
Yeah.
out.
Okay, let me swing around.
Do we have similar concerns on the starboard bio box?
Either, yeah. There's nothing. No, there's nothing floaty in the starboard box.
Roger that.
In fact, most of the rocks in there are pretty small, so
Pretty sinky.
For this one, I'm gonna ask you to hit sample salvo.
Yeah, okay. Ready?
Yeah, and then change the port rail camera to whatever the other one there is.
That's the,
Yeah,
yeah,
that
there
one.
we go. Okay. Knock it out.
Meredith
not
says
yet.
actively swimming.
Hmm.
So, which one are we gonna go in here, guys? Which box are you gonna want it in?
let's put it in the E,
E.
just to save time. Yeah. No corals in there, right? Is there?
Yeah, there's some fossilized coral in there and a
Okay.
rock.
You gotta
That's all
open
right.
it up.
Yeah, I can go.
All right.
I don't know. If I can't tell fossilized coral from live coral, I need to find a new job.
There's no,
It's probably not fossilized coral anyway. It's probably sponge.
no take backsies on that PhD.
Yeah.
Once you dress up like a
Close
fancy
that
wizard
box
and
a little
hand
bit
it to you.
first, so the little ones are back.
I'm so bummed, yeah, because of
Go ahead,
COVID, I
close
never
a
got to
little.
go to my
Yeah, closing
graduation.
in.
Oh.
So I'm not really a PhD. I'm just pretending.
Keep going,
I feel like we should have a graduation for you.
I even
think
bought I even
so.
bought my whole cap and gown and
One more.
everything. And
Oh my
never went.
gosh.
How's that?
Okay, yeah.
That was 041.
Steve says this will sink, but
It'll
trust
sink.
your. Sometimes it sticks. Oh, nice. Okay, you can close it.
Closing sample tray.
Nice
Good
little
job.
box.
Look at that
Nice.
speed. Look at
Atlanta got the
Nice.
shot. I know, look at that speed. The Atlanta shot of the arm's pretty cool.
Lightning.
All right. In the essence of time after this, we're gonna want to just
Spooky.
go straight.
A race to the north.
Yeah.
Okay.
You can write that down in your
ROV
logbook
race.
when he changes his mind in 5 minutes.
No, no, I won't change my mind. You got to get there.
Okay, you can hit one on the bubble cam for me too, please. And then change that camera
Bridge
back
nav.
when you get a chance.
Can we move
I don't
20
change my mind. I
meters,
just get
040,
complacent
please?
sometimes when I see these
Thank you.
landscapes. Waterscape, landscape,
Seascape.
seascape.
On
my save on the cucumber.
Yeah. Is that the one that we put in there, or did they put another one in there after that?
I think the one we grabbed was pretty rigid.
Yeah.
Whoa, hello.
Metalgorgia.
I think, are there 3 sea cucumbers in the forward bio box? I think.
I don't
party.
know.
A lot.
Lots of sea cucumbers.
Ruby crinoid.
Beautiful.
Gonna need some peanut butter for all that jelly, though.
Totally.
No, he's on a roll. Oh no.
Is that a Metalgorgia?
That is
Great work, great way to bring it back to the science.
Sorry, sorry.
It's like, I ignore your dad joke. I must know about the
Oh
Metallgogria.
shoot, sorry. I didn't hear it. What was
No,
it?
it was perfect.
What was what?
The joke.
Do a snap zoom there.
Exactly.
Dan.
Yeah, bamboo coral. Yeah.
Nice.
Wow.
Here's actually a
Beautiful.
question for Steve from the chat. What's the most memorable sample you've collected so far in your career?
The last one.
The next two.
Most recent.
Okay, you can see that.
The one that hasn't yet been collected.
The great white
Said,
whale.
I'm not digging into my memory bank right now.
So I wasn't there for the collection, but Rob was there and got the blue coral back in 2019.
What?
Oh, that was the best.
And that
That's
was,
my favorite.
It's a fantastic specimen.
Was it blue?
No, it was pink.
Yeah.
I'm not imagining it.
It was super blue, like purple blue. It's my Zoom virtual background for the last 2 years. It's been my Zoom virtual background.
Bridge nav?
I love that one.
But I think my most
You can do
memorable
some longer
collection
moves, Lynette, if you want to make up time.
was
Go
Can
back
we
to
move?
50s.
50 meters, 040, please.
an instance in the Caribbean in 2014. Do you remember spiky crab?
Yes.
Yeah, spiky crab. It's possibly a new species of Neolithodes species who are exploring the Anegada Passage
One of my
in
favorite
2014.
cruises. Yep.
And Mike Philemon was piloting Hercules, and we had some trouble because of the consistency of the strong trade winds at that site. We had some difficulties holding station,
I gave you
and
a
we were
DV,
about
I'll
to
reset.
sample this, possibly new species. And I just remember he did this expert maneuver where, it's spiky crab. Imagine, spiky crab, very long spines, very small body, but he managed to get spiky crab like as we were getting pulled off station because we were traveling that way and the crab was here. Managed to scoop it up and toss it into the box. The crab got airtime into the box and closed the box.
Wow.
Uh,
And the crowd went wild.
and and everyone was thrilled.
Cool.
Oh, that was great. That was a really memorable cruise. Yeah, these 7 seamounts stretched in the Anegada Passage, like right in this super popular waterway, one of the big deep water channels to come in and out of the Caribbean, into the Atlantic. And one of my distinct memories from that was finding the little cocktail swords that are the like toothpicks that look like little pirate swords. That were like clearly someone had been having hors d'oeuvres and like throwing these off the side of whatever vessel they were on. And we were just like all caught up in the storytelling of like, we've never seen these seamounts before, it's brand new. And it was like cocktail sword.
I remember a lot of alcohol bottles tossed off of cruise ships in that area. Lots of marine debris.
Think Herc will fit?
Only
Nice
one way to
little
find
canyon.
out.
That was 14,
There
right?
is only one way to
14.
find out.
Look at that.
If you want to go back in the Wayback Machine, check out Nautilus Live and click on the Expedition tab. And you can scroll back to see all the different locations the ship has been season by season. And then click on to any of those expeditions and you can see the teams who were on board, the highlights that were from that dive, the photo albums, everything that was created and written about those expeditions. The other way to do it would be go just below the live feed and click on Steve's bio And within the bio, it has, on the right-hand side, hyperlinks to all the expeditions that people have sailed on. So you can check out kind of the cruise count and the different expeditions, any particular person that you're excited to learn more about has been on.
And if you do the math, it does show that I go home once in a while.
How?
Coral-themed bathroom.
Yeah, what about virtual participation, scientist ashore participation?
I know, we should have like a different section. It's
Yeah.
like a little badge when you
Yeah.
get to, 5 and 10.
What's on the seafloor there, that very flat black looking thing?
Yeah, I was wondering the same.
Brandon?
This,
Where?
Looks like a steak.
Looks
That?
like a purple steak.
What he just circled?
Yep.
Yes. My guess would be a rock that fell in at that angle. And just, yeah, staked into the ground.
Looks man-made to me.
I think it's just the downslope side, sediment free. Although there's another friend in there, another Siphonophore.
Oh, yeah.
Oh yeah, kind of perched with their little anchor tendrils down.
It's just weird the longer you look at it.
It's very weird.
Oh, it's the sand. It's the way the sand's
okay.
forming.
Gotcha. It's a bit of an illusion.
Art.
Oh, is that guy suspended there? Has he got webs?
Yeah, they have little like anchor tendrils.
He might be anchored on both sides there. You can zoom in there?
Yeah.
Is that another sea dandelion?
Yeah,
Cool.
so cool. Oh yeah, you can see a couple tendrils going down.
Yeah, he's anchored.
Mooring lines.
Totally.
Wow.
They look so delicate.
They really are.
How big
Where'd
are
Herc
these
go?
things usually?
Yeah, I'm drifting up above the ridge to your port.
Can you see me?
Yeah, just barely.
Yeah, it's tough to. We're not painting lasers on something in the same
Where am I?
plane, but they're
Down there.
they're little. Would fit in the palm of your hand.
Oh.
Now you're hiding behind the ridge.
Soon to be smaller.
Lost
And
the
then,
plot. Oh,
Steve,
now
is
I get
that
it.
that
Yeah,
dead sponge that we're seeing that are underneath that's underneath that rock?
Yeah, it looks like it.
Huh?
The sonar, that's very scary, yeah.
The rail cams are pretty scary too.
Yes.
Is it crevice or crevasse?
Potato, patata.
Yeah, it's interesting, right? Because there's two spellings too. Like in mountaineering, crevasse is CREVASSE. I don't know if that's like a French versus an English spelling. People with Google, check it out at home. Let us know what the answer is.
What's the name of the little pass where you go through at Endeavor? Where you always have to drag the cables around and go through. A lot like this one.
Yeah.
I can't remember the name of it at the moment.
Starting
Rogers Pass.
Starting to spin me around here.
Roger. I going the wrong way, and I guess I gotta get out of here and come to the north. Roger.
All right, there's some good density here, even on
Yeah.
the tops.
Oh,
So
wow.
this suggests that there's pretty good flow conditions here for a lot of different critters. So call this moderate density, not high density, but not quite a coral garden.
Have you seen any coral gardens this deep?
Oh yeah, yeah. You can see usually Hemicorallium in the right conditions can form pretty dense gardens.
So what classifies as a
You can
garden
come up
versus
a
a
bit
colony?
now, Gibson.
Well, colony is a single, you know, single individual, which is actually complete. That is a complete contradiction.
Why don't you
but,
come right up?
nevertheless,
Come up
let's go with
10
it.
meters.
So colony is
Up
one,
10.
and a garden.
Come up 10 meters.
There is
and
a
then
proper
spin your
terminology.
vehicle.
It's something like
You want to get that wrap out?
Yeah, we need to now.
2.76 colonies
I'm at
per square
1.5
meter,
now.
according to the official definition in a publication that came out a number of years ago. I could be wrong on that number,
Come
but
up
it's,
to about,
it's something.
But are there intermediary
that should
definitions?
be
You know, do we
enough
have
there.
like
Okay,
coral
taking it out.
grove,
Look
and then we
straight
have
down.
coral garden?
Coral
Yeah, I
bushel.
wish.
Before you
Give me a
look
few clicks here.
I tried to get support going for what was it?
Okay, I'm gonna go
Yeah, keep
clockwise.
looking down a little more, just so I can see the tethers are coming
Was
up.
it Sponge Forest or something
Yeah.
a while ago? No, it was.
I don't know.
No one was
You gotta
having
go
it.
counterclockwise.
I'll sign your
Yeah, I do.
petition.
Yeah.
We
All you
have
have
it.
to do is come out with a paper and call it something, and then give it a
Give it a numeric.
number,
Yeah.
and that's it. I mean, that's
6 sponges
that's
within
facts.
14 hectares.
Yeah.
Oh, science.
I kind of like high density. High density gives me lots of room. Could mean a lot of things.
Yeah, but absolutely, deep sea corals can grow super densely, look like a forest, meters tall, meters long, you know, continuous. They're kind of bundles of different colonies together, or, you know, long, just continuous stretches of different colonies. So those are the kind of things we're looking
You
for.
want to go once more around?
Big
Nope.
objectives of this expedition
It's good.
are just to dive on
Thanks.
these seamounts, understand what the communities look like on the very small selection of the parts of a seamount we get to see within one dive, collect rocks that we feel will be representative of the slopes and will help us tell some about the geologic origin and age of these seamounts, and just better understand what is within the protections of the Pacific Remote Islands Marine National Monument
It's a
here
black
in the
coral
Johnston
there,
unit.
Storapathes, Irrigorgia.
These are some of the deepest dives that have ever been done in this region. So adding really important knowledge to the overall picture of what the deep sea looks like here.
So what makes the black coral have the black spine? Is it the way they consume the detritus and nutrients, or?
The black corals produce a skeleton that's made out of protein, versus the corals, which is calcium carbonate, tend to be lighter, typically in color. But yeah, protein is dark black in this case.
So it's what the skeleton's made out of.
Yep. Fun fact, black corals, the name, scientific name for black corals is Antipatharia, and in antiquity, it was thought that black corals would help fight disease, so they kind of get their name from the anti, against,
PATH, yeah,
Ah.
and against disease.
Oh.
So they were often administered as kind of like a rudimentary drug.
That's fascinating.
It's really cool, yeah.
Wow.
Like as a soup, or how do you administer a black coral?
How do you administer black coral? Sounds
Probably
painful.
ground up, I would
Probably-
imagine.
Yeah. Paste.
Did it work?
Guacamole.
Black coral tea.
Did it work?
Obviously somebody made it, wrote
Yeah,
it
I
down,
guess they kept
the
doing
name
it.
around.
Somebody also wrote down that mercury prevented aging, so.
Yeah.
Yeah.
Snake oil, black coral.
Take it with a grain of
Hey,
salt.
50% is great in baseball.
That's your phrase, isn't it? Unless you're a pitcher, then it's not very good.
Yeah, not so good.
Oh,
Really, for
black coral is also the official state gem of Hawaii.
wow.
Oh, a state gem.
Also an animal.
Also an animal.
That presents a conundrum.
A gem animal.
Yeah.
What is the state animal of Hawaii?
Black coral.
Hope not.
Guessing it's something
It's a
else.
bit of a conflict of interest.
Oh, there's many state animals in Hawaii, I think.
Yeah. Let's see.
I believe their fish is the mahi-mahi.
I know.
Isn't it the one with the
Google
really
tell
long
us.
name?
The Humuhumunukunukuapua.
Yes, that one.
Yeah, Rhyncanthus rectangulus, I think, is the scientific term for it. And I'm not sure what the common name for it, though, is.
It's a nice shot.
Fish.
The state mammal is the Hawaiian monk seal.
Oh.
The state tree is the ohia tree.
Shrimp.
The state flower is the yellow hibiscus. State bird is the nene goose.
I love nene goose.
Yeah. State land mammal is the Hawaiian hoary bat.
Ah.
The state of Idaho has a state fossil, which makes me very proud of my home state.
What is it?
It is the Hagerman horse. It is a
Ooh.
zebra-ish animal that was,
Or are those
Found
horses
in-
that were closer to the ground?
yeah,
Yeah.
a little more compact.
Yeah. It's because there weren't many plains, I think, when they existed.
Oh, very nice.
So they wanted to be closer to the bushes.
Can
Mm-hmm.
you take a
Gotcha.
photo?
Oh
go
my
on.
Nice
God.
bamboo coral. This
Wow.
is
That's beautiful.
Keratoisid.
What is it?
It's a bamboo coral in the family Keratoisididae. And if we can get a half zoom, we can figure out how it's branching and maybe
Yeah,
refine
go ahead,
our
Dan.
ID a bit better.
This is the biggest coral we've seen all watch.
So it's a node brancher.
Tell me what you're looking at to make this ID.
Yeah, we're looking at those dark bands that you see. You have to look closely because this one's pigmented, so you can see kind of these dark lines in the skeleton.
Can do.
And if a branch comes off the node, that tells us if it's one group. If it comes off between the nodes, it's another group. Well, it could be among many groups, but there's two main clades for that are based very strongly off a branching pattern. Let's
It's a
see here.
beautiful color. You can sense the scale. Those those green lasers are 10 centimeters apart, or about the width of your hand. So you can start, imagining, counting out there how wide this is.
I think it's over 1 meter tall.
Yeah.
Yeah, about.
Those cool crinoids in the back, the feather stars, purple and kind of cream colored. Oh, there's a yellow one back there too.
Good shot there of it.
So this could be a J-clade bamboo, which is a nodal brancher. They usually get quite large.
Is there any reason why we see this sort of like an orangish, yellowish coloration, and then we see this bone white that immediately cuts off? Is there any reason for that?
That one has no polyps. Yeah, the polyps are dead off of that portion. No predators in sight on these, though. All right. Yeah.
So does that just mean it was a disease or it was damaged some other way?
Difficult to tell. Yeah. Difficult to tell.
Swinging around in Atlanta.
It's probably something
110.
that grazed the coral. So a predator or something like a snail or a sea star or a sea urchin or something like that. Even there's a type of shellless mollusk called Aplecophoran that eats corals like this. And you often find them around the base of the colony or sometimes wrapped around, coiled around the branches. But over time, they will regrow the coral if...
Trying my own active heave comp over here.
Zoom out a bit.
Yep. These, yeah, there's really cool...
It's not working very well.
With this clay, there's really cool defense mechanisms it has sometimes against anemones or ring anemones.
DSC there, did you, Steve?
See what?
Did you get a DSC, did you?
Yeah.
I got several.
That's one from the other side there.
Yeah, gotcha.
Beautiful. Just beautiful.
So if there's an anemone that settles, for example, on the branch somewhere, sometimes what these corals can do is send out like multiple little smaller branches and kind of encompass and surround and eventually calcify over that that animal. So it's kind of like a defense.
It's a super defense strategy.
Nice J-clade. Maybe genus Jasonisis.
That's gorgeous. Look at that Atalanta view.
Oh yeah.
For some scale.
All right.
Oh, so this coral is actually pretty big.
Right
Like a meter wide.
Yeah.
At least a meter tall. Bigger.
And I usually think about how big some of these things down here are, or even get to be.
Yeah, for scale, the height of Hercules is what, 2 meters?
Yeah. Well over a meter.
So that's about waist high for the average person.
Oh. Yeah, at least.
Might be even taller than that.
I'm going to take my turn out in a dramatic fashion here.
I like that.
I'm going to take us back to state fossil. Lewiston, Idaho is giving a shout out. So what's up, home state of Idaho? Lewiston, Idaho is a seaport due to the dams on the Columbia River. Fun fact. So I guess I can't say I come from a state that has no ocean. we claim a seaport.
A, singular?
Yep, one.
We're shouting state fossils, then Nevada has one too, called the ichthyosaur.
Someone wrote
somebody
that
in chat,
in. You've got a Nevada friend writing
Okay,
in.
yeah,
This is awesome.
that's my favorite dinosaur. Yeah, it's a marine dinosaur actually.
At what point does a fossil become a rock, or are all fossils rocks?
Okay, you'll have to take the other half of the turn out there.
I don't.
But I'll come under you first.
Okay, roger that.
I don't know if I'd call a
Just
fossil
be
a rock,
mindful of
I
which way you
guess.
turn there, so
I
we
mean,
get the
it's
turn out.
encased
Yeah,
within a rock.
I won't turn yet.
Yeah, it's all remineralized, but
Right.
does that make it a rock?
I'm going to
Yeah,
come
I
around
suppose it does,
counterclockwise.
It's
You want to look at
special
the little one
category.
here, Steve?
No, we're all set here.
Right.
Yeah, I think it's same as or similar to the previous one.
It's a nice DSC shot though. Nice.
Yeah, nice diversity on this
Good
rock.
one.
I was moving pretty fast. It probably won't be. Might be
looks
a bit
good.
blurred.
Looks good.
Oh yeah, very nice.
Yes.
Oh, nice flyby. Okay, I'm
Looks
taking
super
another
good.
turn out now.
Oh, wait, I'll come under you.
Okay.
You'll have to come up a bit.
Yep. Coming up.
All right, 1,700 meters. You're much improved to 1,900 meters.
Yeah.
I have faith. We'll get there.
So
Okay.
Lynette, what is
45
the
minutes.
shallowest point on this seamount? Are we
The
going-
shallowest point, period, or the shallowest point that we are going to?
Okay.
I want to
you
know
might wanna
both.
Come up another.
Please.
A couple meters here.
Yeah, and then
Yeah, I'm watching that ridge just to my aft.
yeah, you can spin around now.
Okay, coming around.
Oh, that might be a sea cucumber.
It's always cool to spin around while you can see her.
Yeah. Agreed.
Bonk, watch out for that rock.
Yeah.
So here's a biologist question asking a geology.
Yeah, you'll have to come up.
Biologist
Coming up.
asking a geology question. Are all minerals rocks?
So a rock is a collection of minerals, by definition.
Okay.
So one mineral, would it be considered a rock? That's
So are
arguable.
So
It should be
then the
good there.
question is,
You're
are
up above the
all
ridge.
fossils one
Gotcha.
mineral, or are they
Right, that is
different
the
Well,
types of
kind
minerals?
of, no.
See, now we're
yeah.
getting to what
Oh my
defines a
God.
mineral. Is it just a single crystalline structure?
Steep.
Like,
Yeah.
is it one branch of calcium carbonate a mineral?
It's great. You're on channel 3. Let's have this discussion.
The heated debate of the van.
Stay tuned. You're going to want to know the answer to this because it's going to be a Final Jeopardy question one day.
Hold the ship there for a minute.
Is a hot dog a sandwich?
Yes, I think. I'd be more confident about that.
Okay.
But is cereal a soup?
It depends if you're eating raw cereal or cereal.
Cereal.
What's
cereal,
raw
with milk.
cereal?
Yeah, what's raw cereal?
Just cereal.
Keep coming up all
Okay,
the time.
like
What's
is there
cereal
milk in
cereal?
it?
Yeah, without milk.
We don't know
Okay.
how big the
Yeah.
cliff is, so.
Yeah.
Mm. Just raw cereal. Turns out that was actually a really good description. I just wasn't ready.
I don't know how else to describe it.
Yeah, just a bowl with
Dry.
cereal or a bowl of
Unprepared.
cereal.
There we go. Dry cereal.
Unprepared, yeah.
Unprepared cereal.
Okay, you can
Yeah.
get her moving
Incomplete
again.
cereal.
I'm happy now. Sometimes those will
Like
go
your
on
cereal
for
soggy
a
or
long way
crunchy?
too, you
Or
know,
unprepared.
Get
longer
up.
than we can get up.
Yeah.
But we're good.
That's gonna be the next warm-up question. Just wait. Favorite cereal.
What's
I'm
your
ready for
favorite
it.
type?
This is how you divide
Like
an
how his answer
exploration
was just
vessel.
no. I don't even know what it was
Well, I've only
about.
seen Rice Krispies and Raisin Bran in the galley.
Wait, what? Raisin bran?
Is that a thing here?
Yeah.
Yeah.
I'll be careful what I say. People laugh at the things I say from Canada.
You're doing great. Don't be shy.
Totally raisin
I love
because we
learning new
outnumber you.
words.
What did I say the other day? Zap straps.
Zap straps.
I think
What
we
are
call them
those?
zip ties or tie wraps here.
And jubes?
Right. Jujubes.
What?
Like the little gummies.
Jujubes.
Jujubes. The candy?
Yeah.
Like a gummy
The
snack?
candy, yeah, that's
they're
right.
like dots, but super, super small and
No,
disgusting.
they're big.
Oh,
Yeah, they're big.
so not jujubes.
No, yeah, no.
Wait, are they? I don't know. I don't want to get back into this.
Anyways,
Can lift
We're
to
having
your left a
cultural
little bit. I'm
exchange.
gonna come around this rock here.
The mixing of cultures.
Gibson, I'm gonna need some Canadian candy here soon.
it seems to be all about the ocean.
It is all about the ocean.
What about the ocean, eh? There's actually a
You're
question.
not alone, Gibson. Folks are watching in Canada.
Awesome.
Hi to our viewers.
Hello,
in-
everybody.
Yeah, hi to everyone in Australia, the United States, Germany, Denmark, Finland,
It's getting
UK, Hong Kong, Korea.
Wow.
Waking up over in Europe. Good morning.
Good morning.
To everyone else, good evening, and to everyone else, good night.
It is 8:15 p.m. here on the ship.
Here's a question from chat for the ROV pilots.
Sorry.
If Hercules had a microphone, what sound would we hear, if any?
Can you demonstrate the sound for us?
Please give it audio representation.
I really do want to be able to just record it on my phone so we could play it back. Look at all of them.
We have had microphones on vehicles. You hear the hydraulic pump, and
Nice little
but if you get fancy,
spot.
there's some software to filter that out. It's somewhat effective. What's really useful for us as a pilot is you hear when you bump into something,
And if you
like with the
listen-
manipulator when you're tapping, like someone knocking on your door.
Oh, that star's jumping.
Oh yeah, wow. What a shot.
It's a bunch of crinoids on a Walteria, two of my favorites in one spot.
Wow.
It's very pretty.
Amazing.
That's just for you, Lynette.
Lynette's
I
summit.
appreciate it. Thank you, Ocean.
Love it.
So would this be classified as a garden?
Yeah, you're getting there. You're getting close. It's
Ooh,
definitely
and
got a diversity component that
Look at the
would
spiral.
suggest that. Yeah.
What's that one's name?
Craig.
Which one? Oh, this one here?
Yes.
All right.
Ooh.
So I believe that is, that one right here in the lower right is Rhoda and Irigorgia, I think. But I need to get closer to
Starburst.
get a look.
Rodana Irrigorgia.
Yeah, that one might be different species.
Not just regular Irrigorgia.
Before we leave this area, we'll want to get close to the middle of this little high density area and then trigger our last Niskin too.
Go ahead, do that.
It is a Irigorgia, it's Chrysogorgia, but I can't tell
zoom in a
if it's
little bit
Rhodana
there, Foster.
Irigorgia or something else.
Wow.
Wow.
So it's not Rhodana Irigorgia. What it might be is actually a species called Irigorgia bella, which has a tighter spiral like this one does. Sometimes the branches are larger, but it's a very nice, fully intact colony. Not Rhodana Irigorgia. Its axis is not really all that wavy. It's more of a tight coiled helix. So possibly Irigorgia bella. All right, I think we're ready for that Niskin.
Roger.
There are other species of Irigorgia, so it could be something else, but at this depth, you really only see a couple of them. Magnus borealis, which has very wide coils, which is most of what we've seen so far, and then Bella, which usually has tighter coils, but coils, take a look here.
Question in chat of what time zone is Nautilus using watches for? We are using the Hawaiian Standard Time.
That's right. Yeah, we are.
And you hit 5
I was
on
about
the,
to say we are only 800 miles
5 on the
from
bubble.
Hawaii, which in big Pacific and time zone standards is relatively close.
Still, it's all open ocean here. There's not a lot of land you can see.
We're about 150 miles from Johnston Atoll.
Nautilus, or,
Pretty far away and in really deep water, with deep water surrounding us kind of in every direction. So.
I was trying to get the vehicle to float.
If you want to learn more about the history of Johnston Atoll and the National Wildlife Refuge that's there now, you can check out some great info on NOAA Fisheries and the U.S. Fish and Wildlife websites, which are both linked in our latest blog, which if you are over at Nautilus Live and click the gallery tab or the status update right above the video feed, you can check out that blog to learn a little bit more about this area and its history and how the work that we're doing is going to support the conservation of this area.
Yeah. Happy to trigger it here, Steve?
Ready? Yep,
Not
ready.
too high?
Yeah, I think that's okay.
Okay, here we go.
All right, you got it.
Nice.
What sample number was that?
042.
42, okay. You're doing pretty good for sampling.
Pretty well.
What is like an average sample number on a 24-hour dive? Is there such a thing, or is it highly variable?
Could never
Uh,
have too many.
On a typical dive, it doesn't have to be 24 hours. Between 15 and 25 is typical. Yeah. I think we took at least that many yesterday.
You're right, Lynette. Those are awesome.
Right?
Hit one on bubble for me.
One on bubble. But your screen's HiPAC survey right there? Is that what that's called? Okay.
Coming at you.
Yeah, let me come up a little bit here.
Depends if Steve wants to look at this rock or not.
Let's keep going.
Roger.
I told myself I'm not going to change my mind. No, I mean, most of the stuff we can get a good handle on. You know, we've got Victorgorgia, Hemicorallium, paragorgia in the background. Most of these we have collections for in this area, either at museums or in collections from other cruises. This is another type of bamboo coral here, different kind of nodal brancher. In the foreground, white polyps, larger bubblegum.
Shallow water stuff, eh, Steve? Put on your scuba tank and see this stuff.
Yeah, yeah.
Free dive.
Unfortunately, I'm 70 million years too late.
What's that?
That's something
Oh,
really
what's
cool.
floating
Time
there?
It's
machine.
floating.
Fish. You want to see
Yeah,
the
so
fish?
this is kind of typical for what we see around
Yay.
Oh.
the crest of these seamounts. There's like it's almost like a
Oh, wow.
large boulders that have these high density communities right around the top of the crest. Oh, I think we've got a
Tether's
cuskeel
coming
there.
tight here.
Yeah, I'll come back towards you.
Drop down a little bit. Oh,
So is that
Irrigorgia.
Irrigorgia or is that Irrigorgia?
That's Irrigorgia,
Okay.
probably Irrigorgia bella or something in the middle. The one in the background there, that's more magnispiralis. A very wide helix.
Ooh, the big one.
Yeah. This is probably bella in the foreground here.
These are my favorite. We're coming up on 4th of July, so Irrigorgia.
Oh, yeah,
and
what are we
then
gonna
the
do?
big. Sorry, which did you just say?
You can
Magnispiralis.
Magnispiralis.
you can pull your you can put ROV nav in where the Argus is there,
Are we gonna
or
have
you
a
can
4th of
you
July
can put it where you
barbecue?
normally have it, where the DSC
Maybe.
is.
Yeah, maybe.
I hope so.
I want to leave the DSC up too. I can put the DS here on right.
This will be my second 4th of July in a
No,
row
I
at
need
sea.
that.
Okay.
Need that
That's either
sonar.
really cool or really sad.
I'm trying to
Don't
figure.
don't let me tell you how many kids' birthdays I've missed.
Oh no. Oh no.
We always make it up.
It's my second. I guess it wasn't last year. Like 2 in the last 3 4th of Julys, I guess.
I don't know if I can get around this one. You might have to
Oh yeah,
great.
there's a
let me yank
predator of the
Oh, you're
base of
yanking
this
already.
colony.
Auto headings off for telltale.
Nice
Sorry, what did you
Tony
look at
asteroid
before?
star.
you. Yeah, I don't quite have enough leash to get around it. Sorry. Maybe we can do it. Come on, Herc, you can do it.
And track the.
All right. Lean this way. Rip the joystick out of the console. Okay. Is it gonna do it? It's not gonna be pretty. I'm chickening out and coming up above it in case I get yanked. I don't want to get yanked into it.
Yeah, we're just above it in the butt can there.
Yeah, Raj.
Nice big bamboos. Typical for these
seamount summit communities.
Towards you.
Okay.
Not
Probably
necessarily
lots
because
more
of the
to see.
summit. I think it's a combination of factors.
Oh,
The
wow.
substrate is
Look at
probably
that.
ideal here. The flow
Wow.
conditions are probably ideal.
Irrigorgia.
Substrate is very stable.
There we are. Hello, Herc.
Metalgorgia.
Firework critters, as Kevin says.
Oh, my hands are getting tired.
Okay. At some point we got to go the way you're going.
Yeah, we can go whenever.
Yeah, we're going.
I do want to. The purpose is
Get somewhere.
well, it's to push into the summit of the seamount a little bit to see kind of what the variability is as we move further,
Roger.
both upslope and onto the Guyot summit. Still fairly rocky up here. I thought it would turn to sediment, but there's a lot of ridges and things.
Yeah, it's a nice boulder field there.
Oh, Megan, you were asking a while back about the elevation of the summit.
Mm-hmm.
And we have less than 100 meters to go in elevation until we reach the peak at 1,647 meters depth.
All right.
So if you were to do an orbit pass over one of these mounds, would you be able to pick up the corals?
I think so.
Just out of curiosity, keep that in the back of my mind.
You
for
can,
other-
so
Yep.
that thing has adjustable range and gain and all that stuff. We were using it to, we had a 1 meter stick laying in the mud on ONC. So when we were flying around making 3D point clouds of pretty small objects we were placing on the seabed. So he was picking up that 1 meter stick, I believe. You'd have to ask Chris how, what that detail is. I know it picks up bubble streams for methane hydrates, so it seems like if it picks up bubbles, it could get corals. Okay, I'm out in front again. We have a little more time to look at stuff now.
Can you zoom on this over here?
Sure.
Do some checks, Dan.
Go ahead. You can zoom in there.
Sure.
Perfect, thanks.
We have seen this at other sites in the region, not necessarily Johnson Atoll, but we saw this in expeditions last year, as earlier this year. I think we were between heteropathies or parantipathes, but I know this was collected. I just wanted to get a good imagery of it since it's a fairly large colony. Could be useful for identification purposes.
So Steve, is there any specific type of either coral, sea cucumber, crinoid, anything like that that's more commonly found around Johnson Atoll specifically?
Tough question to ask, because I would then say, like, what depth are you talking about? Because you might have different answers based on the depths. The deep sea is really interesting, because even though, if you think about the closest analog, which is like mountains, right? There are different zonation patterns up a mountain. that effect is magnified, I think, when you go into the deep sea. Like, you can still have, plants and things and high up in the mountains, but, you might not have trees, or you might have bushes or low grasses or something like that. But the stressors that animals experience as you increase in depth, and the oceanography changes with increasing water masses as you go deeper, it's much more stressful for organisms to adapt and evolve to that. So what you find is that sometimes, your geographic similarities, so latitude, longitude, over very large areas, from the equator up to Hawaii, might be very similar species ranges and species compositions, but even 500 meters shallower or deeper from within one area within Johnston Atoll, say, you can have totally different assemblages because the environment is so much more different. 500 meters or 1,000 meters vertical compared to thousands of kilometers horizontal.
Anything in particular here, Steve? We got time again?
I'm
Still up
looking.
front.
Nothing in particular. I guess.
Roger.
Yeah, could do a little flyby maybe.
Time are we supposed to have this ROV full of samples on deck? Still midnight, is it?
We are going to be off bottom in about 25 minutes.
Really?
It's 1935. Gauges look good.
Copy. Thank you.
Actually, that was a Zoanthid colony there. So this is Zoanthids.
Oh, yeah. Zoom in there if you want, Dan.
Sure.
Oh, no, it actually, is it?
Kind of looks like Chrysogorgia.
It's got a it's a bramble of, Zoanthids and Chrysogorgia, both. This is probably a very large colony of Chryseus or stellata. Yeah. Okay,
Beautiful.
thanks for that.
I'm happy with the speed there. But you can go 0.3 if you want. Sure. Got 25 minutes to.
Yeah, let's just see how far this goes into the summit.
Yeah, if we're not stopping to take samples.
Well, you know me, we'll wait till about 5 or 10 minutes before we have to come off bottom, and we'll stop and take 3 or 4 samples.
Yeah, I like it.
And we'll still be off with 30 seconds to spare.
Could be
You like
sampling
your
right
style.
while the winch is coming all the way to the last
Yeah,
second.
it wouldn't be a successful dive if you weren't
Pushing
using
the
every
envelope.
minute of the bottom time.
Yeah.
I agree.
Any thoughts, impressions about what the current is doing up here?
No, not really. We could do a drip test, and it's not like wicked blowing the tether over to one side or anything like that. Yeah, this will be more fun.
Dunk.
Boom.
If I let go of the stick. Stop. Herc, why are you doing that? Oh, sea bias. Can't hardly see the particles doing anything. That didn't tell me much. I didn't get a sense of what it's doing from that.
All right.
Try this one. Just let it push Herc for a minute. See what happens. Good, just sitting there, not going anywhere.
Yeah. Sluggish.
Usually by this time I would see some speed over ground there on the ROV computer. Not seeing anything. I wonder if it's tidal with the like the sand waves, you know?
Yeah. Could be.
Seasonal.
If I get the timing just right, I can get the tilts onto some pseudo-active heave comp on Atalanta. Ready? Oh, no. Too
Yeah,
slow.
I get Herc's now moving at 0.1 knot.
Lightning fast.
It's moving at 0.1 knot at 295.
Southeast towards the northwest.
Yeah, now it's swinging around at 300-ish. Still that same really slow speed. Yeah, the course over ground and speed over ground. So Herc's just kind of holding its heading and its depth. And after a while, the current will start pushing it where it wants to go. 300.1.
Well, that's interesting. Yeah, we had kind of, the models suggested kind of a gentle southward flow on this side of the seamount, but it's interesting. I think, you know, seamounts are sometimes known to have some rotational flows, so there could be some like a clockwise flow around the summit of the seamount, or it could be something tidal, you know, and we're just kind of in the middle of that changeover.
We could do, you know, get a little closer to the seabed and get lined up with the ripples here. And it could just be a little local thing.
Well,
like
yeah,
you say.
the ripples are oriented.
Yeah, not what we were suggesting there, right?
Yeah, they're oriented parallel to the apparent flow, which is not typical.
That slow movement there could be attributed to the tug on Herc's tether from Atlanta.
Mm-hmm.
That's the same deal there. So I'm, while the computer is holding heading and depth.
We could also be stuck in, we're surrounded by boulders, so these ripples might not be realistic indicators of what's going on in the larger environment too. It could be channelization going on.
Now it's drifting at 22 degrees. So still northish. And that's, yeah, kind of also the direction we're moving, so I don't know. In any case, not a significant.
Well, that, you know, we can use the biology too. That crinoid in front of us is sticking straight up. That's not typical. Usually they're oriented one way or the other.
Yeah.
So that's a good indicator that this might be more of what is consistently experienced up here. Kind of maybe a very gentle northward flow, something like that.
Here's a good example of how much better the ROV flies when I take my hands off of the controls. Sometimes forget that.
Oh yeah, big Brinoid colonies here. Most of them look like Caltrophora fans. Oh yeah, what's this one over here to the left, the far left? Oops.
Far left.
This one? It's a big something.
Wow. That's a lot of coral.
Can you zoom in on that?
Sure, yeah, go ahead.
Yep.
It's not another Chrysogorgia, is it?
No, this is a bamboo coral.
Nice.
Let me back off and come down. Just bumped it and it caused it to spawn.
We have another chat question for all of the cabin. What is the worst weather you've experienced during an expedition?
I'd say my first day on the open ocean felt pretty bad.
Yep, right there.
All right, this is a keratoisis or ectomysis D2 clade. Different branching at the internodes, rather. So you'll notice the branch points are arising between the nodes. Different than what we've seen in the past.
Herc is like
Probably
Ferdinand smelling the
ectomysis
flowers.
based on the polyp morphology.
Up to Steve.
What's up?
Do you want to ship's move?
Yeah, we're not going to sample this.
Okay.
Yeah, you got enough there, do you, Steve?
Yeah, I think we got enough for an ID, yeah.
Right.
Maybe something more too.
Yeah, there's a hitchhiker.
A sample anyways.
Well, if that sticks around before we come off bottom, we can stick that in the box.
I could grab it on the fly if you want. You know where it came from.
Yep. Yeah, if you want to do that, sure. We'll have to do some fancy back calculation.
What's right here? If you want to drop
We
it.
did get a
If you want to
sample.
drop a waypoint before that.
There's a bit of a coral stuck on the front
we're
porch
gonna
here
grab this
from
hitchhiker.
that,
and-
bamboo.
Okay.
So
oh, okay.
we're not near the colony anymore, but we're going to stow it in the starboard box anyway.
Got it.
Maybe here. I think there's room here.
There should be. It's just
Yeah.
nodules and an urchin.
Yep.
Cool.
Either of them.
that
But
better?
definitely one of the bigger outer
You
boxes.
want the bubble on the porch or the craft?
You're going to put it back on the porch there.
Come on over.
But what we'll have to do to get those sample photos is go back and
look through the raw files because we're not around there anymore to take photos of the collection as it's occurring. So we'll have to just make a note in the sample sheet that we have to look up the raw photos from the time.
And is this another type of bamboo coral that we're seeing?
Yeah, it's a bamboo coral, D2 clade. And the time we were there was like 05:45-ish.
Well done, Dan.
Wow.
Expert.
Nice.
You want that in a box.
All right.
Oh no.
Oh.
Oh well.
Well, we got the important piece there. Make a note too that has a crinoid attached to it, yellow crinoid.
You want to do the sample salvo there?
Yeah, sample salvo.
It's probably, it will sink, but it's probably more fragile than you think. Either of the outer boxes is appropriate.
Right. That's not the sample salvo there. You have to hit it again. It did a weird thing.
Better?
Yeah, and then change that other camera. Sorry, Steve, what box are we going to go for?
Starboard bio box F.
Roger. It's going to take my auto depth
Maybe
off.
E or F.
Load up here so the...
Either E or F works fine.
Yeah.
Yeah.
E has more room. There's a big old scoop in F.
Roger.
You can cut it if you need to. The two corals that are in there, not aside from the fossil sponge, the two corals that are in there are very easy to tell apart. So it's okay to put two white corals together if you know which families they belong to.
Okay.
So we can tell those apart when we get the back on deck.
Nice.
Very shortly.
Very shortly.
Exciting.
Thanks everybody
Okay.
writing in. We
Close
did have
up.
a security certificate error on Nautilus Live, but I think all is repaired now. So thanks for sticking with us. We're nearing the end of this dive, but our plan is to be back in the water in just a few hours after recovery, probably around 6 hours after we pull the vehicles back up. We'll keep the status updated on Nautilus Live with the latest. But I have to transit over to our next seamount target after recovering this dive.
Sure. You want to unsample salvo there for me?
That looks odd. Is that right?
That looks right. You just, yeah, you might have to hit it again. It did some weirdness with it.
There we go.
I'm not sure what's up with that. You can hit one on the bubble camera too.
Yep, coming down. Where are you?
How's our... See, you must turn the funky way again.
Hey.
Just take that turn out while we're
Yep, taking it out.
underneath you.
All right, we're going to do a watch change. Thanks so much for being with us. Keep your questions coming for the next team.
SPL check. 1, 2, 3, 4. Check, check, check.
Bless you.
Just like making sure that the bridge is in a good location, and then depending on it kind of depends on sort of pilot's choice. Some people like to move the ship forward at a really slow speed, just to get the vehicles like kind of tracked out behind the ship. But yeah, check in with your pilot and see what they want. Yeah, and then just some communications with the bridge. And then once you're at 300 meters, you're going to switch over to radio, and it's totally fine. And you're not going to talk on this. Oh, really? Like, yeah. Yep. It should be good. You can turn the volume up a little bit. There's also... Sorry. Yeah, sometimes it can be hard to hear, so I like to keep that on. But I usually turn off, like, listening to SPL and, like, people that I don't need to listen to, like, when we're recovering, because it's just, like, chaos in your ear. Yeah. Yeah, and then, they'll just be coming
Hey Dave, can we zoom in, please?
You betcha. Time to go to work. Whoops, sorry about that. Brittle stars are bailing out.
Oh.
Okay, we'll come wide, please. So Rob, did we confirm an on-deck, preferred on-deck time? Was it 10?
I don't know. Emma, did they get that with you?
That's what. Oh. Oops.
10 question mark.
Yeah, that's what I heard. Was 10 from
Okay.
Steve and Allison
Okay.
last. Yeah.
Yeah, I asked Steve. He said that the navigator would tell us.
We're
I think
almost at
it's
the
like
top of the
133
seamount, right?
minutes or something.
Yeah, I think we're on top of the
We're on top now.
geo.
That's not right.
Oh,
Yeah, so...
At 15
What's the
meters a minute.
difference?
I did the calculation.
Flat top
oh, it's
carbonate
17. Yeah, it's
platform.
like 113 or so, or 113
So it's like
minutes
a
or something like
mesa.
that.
So,
150.
like, yeah, like
Exactly.
2 hours, yeah.
but it's like an underwater
2
mesa.
hours.
It's an underwater
Right,
mesa.
So,
Huh.
yeah, if we want to be on deck at 10, we got to start up pretty soon. If not, I'm going to keep looking at
Is that a
stuff.
Let's
basket
keep looking at
star?
stuff.
No, that's one of those...
Bazingid? Brazingid? Brazingid? Sorry. Not making a joke.
starting off with a bang.
Crinoid. Stop crinoid. Stop crinoid. Wow, we're seeing more stuff than we did on our last watch.
Wow, how exciting.
Did you see them
Yeah,
when
I've
we're
seen
on the edge there,
Purple.
Kylie?
Purple coral. What was it?
But about an hour ago they had
No.
just gorgeous stuff around the edges.
No.
Lots of corals.
No.
Yeah, that last one, there were a lot of beautiful corals with sea
Yeah.
stars on them, yeah?
Yeah.
What were those corals?
I think there were some primnoids
Yeah.
Oh.
and a lot of bamboo corals.
Look at that. Is that a shadow? Oh, yeah. On the crinoid.
Is that in the water column? Invisible.
Yeah. Could be a polychaete or a
Yeah.
ctenophore, hard to tell.
Oh, that coral right there, it was purple?
Okay, we'll zoom in, please.
Gorgeous.
Oh, wow, look at that.
Neat.
Does it have 2 different color polyps, or are they red polyps and yellow branches? No, there's, what's going on?
I think this one's one of this looks like it's been colonized by a bunch of zoanthids.
Oh.
I think those are zoanthids, and then the purple are the polyps of the coral.
Yeah.
And the brittle stars holding it all together.
Wow.
So that's 3 organisms in one.
That's wild.
That
Yeah, come wide,
is.
please. We are, yeah, right after this cruise. Okay, interesting, yeah.
Um,
Something.
thank you. Are you reading our?
This looks almost exactly like a feature they saw earlier. The scour around the boulders like this. It's pretty neat.
Yeah.
Is that suggestive of high flow?
Yeah, there's a current flowing around there.
Nice.
Nice diversity on these rocks.
Zoom in, please.
Oh, look at that purple one.
Yeah, I don't.
Yeah. It'd be great if you could swing around to port and see that purple one too further up.
You betcha.
Oh.
Well, I'm gonna take a little zoomed in tour here.
Okay.
Oh, there go the brittle star.
Oh.
Just at the end
Oh,
of my
look at
tether.
that.
Did he lose an
Looks
arm?
like that brittle star's missing an arm.
There's
Yeah, I was gonna say it looks like it's missing an arm.
there's your purple hoku.
Oh, that's
believe
pretty.
they can
There's
grow
the
them back.
purple.
Do you know what that purple is?
Wow.
I was trying to look for it.
I am not sure what that idea is. I have not seen that one yet. Let's see.
Did the vessel just stop on its own? It does look like it stopped. Oh, no, I can just ask Rob what he wants to do.
Yeah.
Come wide, please.
Wow, look at all that on the other rock too.
Yeah. Can we turn and get a zoom on that purple again from this side? Is that,
Sure.
Or we just move it along? I don't
Oh,
know
look
if we
at
had a
that
good shot to
anemone.
identify it. Get a nice zoom on that. Yeah. Is this picturable or, here we go. Do you, is that yours? I don't
This
have
isn't the
access to it.
picture.
Oh,
That's
yeah.
the draw cam.
Yeah,
Do
okay.
you want me to do it though?
Yeah, could you?
Okay, we'll zoom in,
Yeah.
please.
It was,
Just at the end of my tether, so it'll be a little bouncy. But
is that healthy, you think, or?
Healthy?
Yeah.
I think so.
Okay. That's nice.
Yeah, I can't really tell. Like, it's odd that we're able to see so much of the skeleton, but it could just be because we're seeing a different color coral.
Yeah, it could be.
I'm not
Or it
familiar with
seems like
seeing this.
The polyps are just like kind of on the sides, or like on the edges, I guess.
Instead of along the axis itself.
And is that maybe a little sponge at the base?
Yeah, that's gorgeous.
Yeah. Found a tube worm too.
Oh, where? Oh, yeah, right there.
It's cool to get so many different animals in one shot.
Yeah.
Oh, come wide, please.
Great.
Okay.
Thanks, Josh.
Roger. So we should probably start coming up.
Okay.
And that's it.
Oh,
Yep.
wow. Thank you.
Hope you enjoyed the show.
You can just say stop. We'll start moving once we get to about 300 meters, and we can start moving the ship.
Man, these ships are going really fast.
All right, I'm just
So
going to
within
start
10
coming up like
minutes.
this, and then we'll just kind of see
find
what
it?
happens. What do you think?
That's what somebody put in the chat. It says.
Stretch out and start coming up.
It kind of looks like that one.
This one right here.
Okay.
Right here.
I will not be an auto heading either.
There's that one.
Just let the winds do what they want.
Yeah,
We'll
click
try
on-
to head a little bit.
I mean,
This
it's
one?
it's in Japanese, but I think that looks like the one we had.
Oh.
Yeah, right?
What's that?
Yeah.
Or this one right here.
Yeah.
Does it give you the name?
I thought I saw Victorgorgia.
Or
Victorgorgia?
Victorgorgia.
Said that. I said that, didn't I?
Did you?
I think you did,
I
yeah.
did, because I remembered on the last trip. I'd be learning
Good
the corals.
job.
The purple ones is like royal color. And this was like Victor. Okay. Anyway,
get
yes.
ya.
Oh, there you go.
Leela
Nice.
trained me pretty good. She'd give me little quizzes during our watch. She'd be like, Kylie, you know that one. What is it? And I'm like, it's the one. It's the one you told me about earlier.
It's that one.
You know.
Oh, somebody saw a sea spider.
Almost
Yeah,
adjustment here.
Oh, yeah, we got a lot of the Victorgorgia.
Yeah, on the money.
Can you just show me that little, I guess I can sort of see it already.
Oh,
That little loopy thing.
you're going to pitch a weight?
Huh? Mm-mm. Put it in the other plug? Okay. Because there's a other plug for your headphones on the second level. See the other circle? Is that where you plugged it in before? Okay. Oh, there, Dave will help you.
Okay, Josh, whenever you're ready, I think we can come to the surface.
Okay, we're on our way.
Roger that.
I'm gonna grab this other one too.
So, aloha mai kako. We have now, we will begin our ascent up to return Hercules back to the ship. And so our next dive, we will have around 3 hours of blue water time. So if you folks have any questions.
I think 2 hours.
2 hours of blue water time. Thank you, Kylie. So if you have any questions, comments, things you want to share, please let us know while we're ascending. Thank you. Aloha.
No, it's all good. Yep.
Video. Can I have one of the winch views on the Mon 1?
You betcha.
Thank you.